Okay, activity number 3 is all about the morphological classification of our bacteria. So this experiment would help us to identify the morphology of the different organisms here, bacteria, that's being listed in your laboratory manual. So just need to identify as to their shape. So basically their shape could actually be a cocci, pag cocci, so that's more spherical, so round shape, or it could also be bacilli, so rod shape or elongated. Or you could also classify them as to their shape as with the spiral na bacteria. Or you could have here other atypical shape of the bacteria. Then another one as to the arrangement, so they could appear here as single, could be in a cluster, could also be in chain, okay, or any other arrangement. And another one would be the gram staining reaction. As for the gram staining reaction, so we could classify our bacteria as either gram positive. So typically, the gram positive bacteria would appear to be blue in color or violet in color. And gram negative bacteria, on the other hand, appears to be red or pink in color. But you could also have here the gram variable. So gram variable are those organisms that could appear as any of that. So usually pag nagdo-drawing tayo, for the gram variable, we tend to use here pencil. For the, usually ang ating mga gram variable are mga spiral na bacteria natin. Okay, so in your laboratory manual, there are different organisms listed on that. So all you need to do is just draw individually on the specified na, okay, na microscopic field, and then try to put also here the total magnification since we are expecting this one as a dried smear. So we tend to identify this different structure under the microscope in the oil immersion objective. So that's why you need to label your microscopic field with 1000x total magnification for that. So make sure that each of the morphologies bacteria is eventually are eventually clearly drawn. You need to draw it in such if that it appears to be like pagbilog, so cocci, bacilli, something like that. Or you could have your other structures present on that. Okay, so that's all for this activity. Okay, thank you.